Hello everyone, hope you all are doing well. So today we are going to do topic 16b of paper 2, that is remaining parts of modes of reproduction. Modes of reproduction. So the first of all we are going to discuss in this topic is micropropagation. Micropropagation. So what is micropropagation? It is the artificial process of producing plant. Artificial process of producing plants vegetatively through tissue culture or cell culture techniques. So it is the artificial process of producing plants vegetatively through tissue culture or cell culture techniques. So what are various methods through which we produce in micropropagation are meristem culture, we have got callus culture, suspension culture, embryo culture and protoplast culture. Etc. So these are the different methods through which we can do micropropagation. Now what are the advantages of micropropagation? Now what are the advantages of micropropagation? First of all, this is an alternative method of vegetative propagation with enhanced multiplication rate. Alternative method of vegetative propagation with enhanced multiplication rate, very high multiplication rate. Here large quantities of identical parts can be obtained. Large quantities of identical parts can be obtained from single plant tissue, in very short time. Shoot multiplication has very short cycle, and each cycle results in logarithmic increase in number of shoots, logarithmic increase in number of shoots. Here small sized propagules can be stored and transported easily, as here the produced propagules are small, so they can be stored and transported very easily. Germplasm stock can be maintained for several years using this technique. Germplasm stock can be maintained for several years using this technique. Production and maintenance of pathogen free plant varieties. We can produce pathogen free plant varieties, helps in obtaining desired sex of plants, helps in obtaining desired sex of plants, desired sex of plants. Moving on, here we can produce millions of plantlets. Here we can produce millions of plantlets in cultural vials. In cultural vials. Genetic uniformity of propagules can be maintained through this. Here we can maintain genetic uniformity of propagules. It is cost-effective process. New variety of species can be propagated. New variety of species can be propagated. A requirement of less space and less human resources. Less space and less human resources. Independent of season and can be carried out anytime. Independent of season and can be carried out anytime. Suppose we need to grow some plant in some certain seasons, but here we can grow that in any season, can be carried out anytime. Assist in regenerating genetic modified cell, assist in regenerating genetically modified cells after protoplast fusion. Often produces healthier plants leading to quicker growth. Produces healthier plants which leads to quicker growth. And it has been used and can be used in plants such as banana, citrus, grapes, lilies, etc. So this was about macro propagation. Now moving on, what is apomixis? Apomixis. So apomixis is development of embryo without sexual fusion. Development of embryo without sexual fusion. Now what are the types of apomixis? First one is gametophytic. Here unfertilized eggs give rise to megametophyte. Unfertilized eggs gives rise to megametophyte. So it is of three types. First one is diplospory. Here mega is for mother cell, that is MMC. In short, mega is for mother cell, gives rise to megametophyte. Second is apospory. Here embryo sac originates from the diploid cell. Embryo sac originates from diploid cell, except MMC. Here from diploid cell, except MMC, either directly by mitosis or after uninterrupted meiosis. Either directly by mitosis or interrupted meiosis. And the third one is androgenesis. C. Androgenesis. Embryo sac originates from generative nucleus. Embryo sac originates from generative nucleus of pollen tube cells of pollen tube cells. The most common mechanism of apomixis in higher plants and the most common method of apomixis in higher plants characterized by presence of multiple embryo sacs. So most common method of apomixis in higher plants is through it's due to the presence of multiple embryo sacs. So it was about androgenesis. Now moving on to second one, that is sporophytic. We have discussed gametophytic. And the second one is sporophytic. So it is also divided into three types. First one is, first of all, in sporophytic, unfertilized cell give rise to directly to an embryo. Here, unfertilized cells. In gametophytic, we know that unfertilized cells give rise to megametophyte, but here unfertilized cells give directly to embryo. And first of all, here is first type in sporophytic apomixis is haploid parthenogenesis. Haploid parthenogenesis. Here embryo developed from egg cell. Second one is haploid apogamy. It is also known as pseudogamy. Here embryo developed from synergids or antipodal cells. Embryo developed from synergids or antipodal cells. Antipodal cells. The third one is adventitious embryony. Adventitious embryony. It is also known as sporophytic budding. Sporophytic budding. Here embryo is directly developed from nucleus or integuments. Embryo is directly developed from nucleus or integuments. So, as the embryo directly develops from nucleus and integuments, there is no production of embryo sac. It can be seen in mango and citrus. Now, what is the role of apomixis in plant breeding? Role of apomixis in plant breeding. First of all, it helps in rapid production of pure lines. Secondly, it helps in maintenance of superior genotypes. Thirdly, it helps in conservation of heterosis. Heterosis is often known as hybrid figure. So this was the role of apomixis, and that was the types of apomixis. Hope you all understand what apomixis are, or what apomixis is, what are its types, and what is its role. Now moving on to the next topic of this session, that is parthenogenesis or parthenocarpy. parthenogenesis or parthenocarpy. It refers to development of embryo from egg cell. It refers to development of embryo from egg cell without fertilization. Without fertilization. It is of two types. First one is haploid parthenocarpy. When embryo develops from haploid egg cell, here embryo develops from haploid egg cell. The plants developed from such embryo are haploid and sterile. So the plants developed from such haploid egg cells are haploid and sterile and haploid, and it is found in Solanum digrum. Now second is diploid parthenocarpy. Here what happens when embryo sac develops without reduction division. Here embryo sac develops without reduction division. You should know what reduction division is. I have taught earlier in meiosis, mitosis. So you should know what reduction division. It is your homework. You will comment what is reduction. It is mitosis or meiosis. Such embryo sac and all cells within it are diploid. So here the embryo sac and all cells are diploid, and it can be seen in grasses such as Taraxacum. Now what are the causes of parthenogenesis? What are the causes of parthenogenesis? First of all, there could be inability of pollen tube to discharge the contents inside embryo sac, Secondly, insufficient attraction between male and female gametes. There could be insufficient attraction between male and female gametes. There could be early degeneration of sperm. Very long style. Slendromatous style. There could be short pollen tube. Slow rate of pollen tube growth. Stimulation of pollination in absence of pollen tube. Stimulation of pollination in absence of pollen tube. There could be incompatibility. I have also discussed incompatibility in detail in previous lectures. You should know this. So these are the causes of parthenogenesis. And how can be parthenogenesis induced? How can we induce parthenogenesis? So parthenogenesis can be induced by following ways. Parthenogenesis can be induced by following ways. Number one is by stimulation of widely related pollens. by stimulation of widely related pollens, by low temperature, by pollinating with X-ray irradiated pollens, pollinating with X-ray irradiated pollens, by treatment with certain chemicals, chemical treatment, for example Velveton, by diploid hybridization, male sterility. Also discussed male sterility in very detail in previous lecture, you should know it. By pollen incompatibility, discuss both mole sterility and incompatibility in detail in the previous lecture, you should know it, very important. Transgenic genes, example Arabidopsis gene, the gene of Arabidopsis. So this is how we can induce parthenogenesis. So that is all for today's session. For any queries, do comment. If you like it, please press the like button. Do share and subscribe. Keep making notes. Keep value adding to your notes. That's all for today. Have a nice day. Thank you.